Welcome to the SB Grid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SB Grid.
Today's SB Grid webinar is from Lorenz Lamm, who is at the Helmholtz AI Institute in Germany. And they're a PhD student, and they've been working on membrane segmentation for cryo-ET data. Now, if any of you have done segmentation in cryo-ET, the way you do it by hand is painstaking and time-consuming and generally maddening. So any tools that can automate this and make it more streamlined would be very welcome. So I've played with this a little bit, and it is quite remarkably good for a pre-trained model out of the box. So hopefully Lorenz will tell us a little bit more about how it works, how it was built, and what you can do with it. So with that, I'll pass over the microphone and off you go.
All right. Yeah, thanks a lot for the introduction. Great to hear that membrane seg already worked for you nicely out of the box. Yeah, today I will talk about Membrane V2, which is our full tool for the analysis of membranes in cryo-ET, where I will put a large focus to membrane seg because that's the main thing that is out there yet. Okay, 1 second. Okay, so the structure of this talk will be first a small introduction into membrane seg and how it actually works, because I think this is an important step to also get a feeling for how to maximize the performance of membrane seg. And in the second part, I will also shortly show you a couple of things that we're also planning to have in Membrane V2, because that's not only going to be membrane segmentations, but also membrane particle localization, and also some statistics that we want to output there. And then in the third part, I will also show you some practical tips on how to maximize the performance of membrane seg and get the best performance out of the segmentations. But first, maybe we can start with a small motivation of how we actually came up with this project. And that was this project together with Ben and Wojciech, where we tried to study a bit more about photosynthesis. And yeah, I mean, that's a process that happens on different levels of organization at the same time. So we have this membrane scale here, where we look at the thylakoid membranes in the chloroplast and see how they are differently organized, because depending on their organization, also their functionality changes. But then at the same time, we're also looking at this single complexes scale. In this case, the scale of the photosystem II proteins that are embedded in these thylakoid membranes, and they are also organized in very specific ways. And the organization of the proteins influences the organization of the membranes and vice versa. So we wanted to study both of them at the same time in cryo-ET. And for that, to be able to do that, we need to have membrane segmentation, so we know where actually the membranes are. And then we also need to have protein locations, so we can analyze these. And in the previous project, it was really like that. So Ben and Wojciech were sitting there manually annotating some stuff. I mean, with some initial segmentations already, but it was still a lot of manual work. And now with some follow-up projects, we tried to scale up these experiments. And I mean, with the tomography setups getting faster and faster, I think it's not possible anymore to segment really everything by hand. So we tried to come up with some automations for these membrane segmentations and the protein locations. And that worked nicely for our projects. And now we're trying to make that also available in the Membrane V2 package, which, where we have this preprint out already, which mainly describes Membrane V2, but we're hoping to update that soon with also the other functionalities. Because the goal of that is to have these 3 different modules, which is first membrane seg, which is already out there, and I think does a decent job in performing some membrane segmentations. And that was really nice because we were collaborating also with the community to gather more and more training data to really be able to show our network how different membranes can actually look. And that's, I think, also a key component of why it performs well. As a second component, we want to have membrane PIC, which is the program that we want to use for the localization of membrane proteins. And there our goal is to design it in a very interactive way where we integrate it into a Pari plugin, and we try to go back and forth very easily between the annotation of proteins and then also the training of a network to localize the proteins. And as a third module of the Membrane V2 environment, we want to have Membrane Stats, which basically takes the outputs of these first two steps of Membrane Seg and Membrane Pick and outputs some statistics based on them. So maybe some easy statistics like protein concentrations or center to center nearest neighbor distances between protein locations. We just want to put out some easy statistics there. But yeah, today I would like to talk mainly about Membrane Seg. So go a bit into detail about that and then explain a bit what we're planning to do with Membrane Pick and Membrane Stats and what is already available there. So Membrane Seg, that was a really nice collaboration here with Alistair and Kevin, where we designed this program that can basically out of the box segment ideally all the tomograms. I mean, it's not working perfectly all the time, but I think it's a good starting point in many cases. So that was our goal, to have really out of the box segmentations for ideally as many tomograms as possible. But then we also wanted to enable easy fine tuning. So in case the model performance is not really satisfying on your tomograms, then we wanted to enable just annotating a couple of patches for training and then fine-tuning the model on your specific problem. And then, of course, if you do that, we highly encourage you to also share your annotations, because in this way, really everybody from the community can benefit from these annotations. Because this is, I think, a huge bottleneck still in cryo-ET and computational methods. We don't really have any ground truth to train proper models with it. So how did we do it? I mean, we used as an architecture the commonly used UNet architecture, which I think is still the architecture to beat. I mean, there are more fancy architectures, of course, like transformers, but I think the choice of this architecture is not as important as really the data that we train it with. So we really try to focus on the quality of the training data to get the best performing model out of it. And with that, it's important to have a large variety in our training data set to really show the model, okay, this is how a membrane can look, but also this is how it can look, and show that also in different tomography setups, so the model sees as many possible membrane appearances as possible. And at the same time, we also try to artificially increase this variability in the training data set by some data augmentation. So we just altered the images and tried to generate some new looking membranes with that. And these are not the fanciest design choices, I think, but I think they are the most important design choices why our model performs so well. So I would like to go a bit into detail of how we actually created these. And for the generation of the training data set, I have to give huge credit also to Simon here, who did a great job of annotating some of these patches. And there we followed this iterative approach. So we started with TomoSegMemTV to create some initial membrane segmentations, which were decent segmentations in many areas, but also had some limitations in other areas. So what we did was extract small patches around regions where it performed well, but also focusing on regions where it had some problems. And we manually corrected those regions and then used all this training data to train our first model of Membrane Seg. But that was not perfect, of course. So what we did in the next step was take this model, predict on some tomograms as well, and extract again the regions where the model had some problems. And corrected that manually again, merged it into our training data set and retrained Membrane Seg. And we did that in a couple of iterations, and the model got better and better with that. And with that, we ended up in a nice variable training data set that showed really different tomography setups as well as different membranes. And one thing that is also important here is that we tried to extract only small patches. In this case, pixel size, extension of 160 pixels. And the reason for that is that we really wanted to make sure that basically each voxel in our training patches is correctly annotated. And I think this is still a lot of work in these small patches, but it's really impossible if you want to try to do that in a full tomogram. There, I think it's impossible to guarantee that each voxel is correctly annotated. And what we did then was this. So this is from one iteration. On the left, you can see one prediction of some membrane seg model. And then we just corrected this. So whenever we saw, okay, there is some membrane that was falsely annotated, then we removed it from the prediction. If there was a membrane missing from the segmentation, we manually drew that in. But then you can also see these green regions, which is our ignore label. And I think this is also very important because sometimes the data is just ambiguous and it's super hard to judge by eye what is really happening. If there is a membrane or, yeah, you can see that there is a membrane, but you can't perfectly delineate it. So in these situations, we try to use that ignore label, meaning that during training of the model, these voxels will not be evaluated by the loss function and will not drive the model in either direction. So we really try to make sure that the model only learns from the membranes that we are also certain about. And yeah, with this strategy, we got a nice training data set, and I think got a nice variability also into the training data. But then to increase this variability, as I said, we also did some data augmentations. So what we did was, just some classical data augmentations like the ones you can see here, where we performed some geometric augmentations like this rotation and zooming. But we also did some intensity augmentations, just altering the image a bit by smoothing or adding noise. But then we also came up with our own more cryo-ET specific data augmentations by corrupting the Fourier space of the patches. So what you can see here is just a patch together with its Fourier transform. And then we altered this Fourier, the image in Fourier space to cause some distortions of it. So one example is that we applied the missing wedge, which is also present in cryo-ET. And we just masked out a bit bigger missing wedge, leading to also some densities in the real space image to disappear. So you can see this membrane here that you can see in the left patch. It's pretty much disappearing after this transform. But we still have the ground truth from the original patch, so we know where the membrane should be, and we try to force the network to still predict the segmentation in these cases. So the intention behind that is that we try to learn to try to make the network learn how to overcome the effects of the missing wedge. What we also did was just rescaling some Fourier intensities. So in this case, we upscaled the lower frequency intensities, meaning to much more pronounced coarse features. So the contrast is just much stronger now in this image. But we can also just do the opposite and upscale the higher frequencies, leading to the noise being much more pronounced. And in this case, it's very hard to see what's actually happening here. So this is making it a bit more tricky for the network, but I think it's a nice way for the model to learn how to deal with the different setups. So this is how we generated our training data and how the data looked that we showed to the model. But yeah, as I also said, we collaborated a lot with the community to gather more training data from different labs. And one thing is that it's sometimes not super consistent among annotators how they annotate the membranes. So this is one example where you can see that even within a single patch, the brush size, for example, of your annotations can vary a bit. So here in this left patch, you can see that it's a very thin segmentation only, whereas here is a much thicker segmentation, even though it should be pretty much the same size of the segmentation. And that is a problem because that is distorting both the training and the evaluation of some evaluation metrics towards these thicker regions. So classically, we use the die score for both training and evaluating our model, and that will just be biased much towards these thicker membrane regions. But this is, of course, not really what we want. So as a synthetic example, what we tried here was just have some ground truth segmentation and some prediction. and compute the dice score between them. I mean, they are agreeing pretty nicely, so we get a dice score of 80%. But now what happens if we just grow these ground truth segmentations and then still compute the dice score is that the dice score is going down, even though I would argue that the segmentation quality is still pretty good in the second and third one. Maybe the fourth one is a bit too exaggerated, but still, I mean, the dice score doesn't show the full picture of the segmentation quality, in my opinion. So we tried to come up with a metric that is more invariant to that. And that is how we developed the surface dice, which maybe you're familiar with the centerline dice. So it's very much inspired by that. So classically, how you would compute the dice score is you have the prediction segmentation, which is this green block here, and the ground truth segmentation, which is the orange block. And then you look at the overlap, and you want to have this overlap as big as possible. Because with this, you will compute the precision, so compute the fraction of the overlap in the prediction, and also compute the recall by computing the fraction of the overlap in the ground truth segmentation. And then once you have these two, you will compute the dice as the harmonic mean between these two. So what you want to have is really maximize the precision and the recall. And that is the case if the overlap basically agrees with the prediction and ground truth. So this is what you tried for the classical dice score. But yeah, as we saw before, this has some problems if you have different membrane thicknesses. So what we do instead is this surface dice, where we don't really compute these overlaps on a pixel level, but instead what we do is that we skeletonize the segmentations. So we take this prediction segmentation and we skeletonize it to a one voxel thick sheet, indicated here by this dark green line. And then we don't compute also the pixel-wise overlap fractions, but we look at these skeletons. And for this prediction skeleton, for the green skeleton, we look at what is the fraction of this green skeleton that lies also within the ground truth, giving us this solid line here, whereas the full line is the dotted line. And then we compute this fraction. So what is the percentage of the skeleton that is basically correct? And the same for the recall. And then we can compute the dice score with that. And the nice thing about this is now that this is, in theory, invariant to the thickness of the membranes. Because no matter how thick the segmentation is to start with, it will always shrink it down to a one voxel thick skeleton. So that should not really change. And that's also what we observed here now. So if we apply that now to these segmentations here, then we can see that the surface dice, is consistently very high. And that makes sense because these segmentations basically overlap pretty nicely. And the nice thing about that is also, I mean, we can use it once as an evaluation metric to really, more objectively judge how good segmentations are without focusing on the thickness of the segmentations. But we can also use it actually as a loss function to optimize our network, so that the network also doesn't really focus on perfectly getting perfect pixel-wise agreement, but it focuses more on the continuity of the membranes. And that's already what I wanted to say about membrane seg and how we did that. So as a summary, it performs pretty well out of the box. And one thing that is also very important for us is that it's hopefully pretty easy to use. So ideally you can use it just with a single command line with just membrane segment and then some arguments behind that. I will go into a bit more detail how to properly use that and what are the different options to use that in the third part of the talk. But if you want to check out some quantitative evaluations of the components that I just showed you, then feel free to take a look at our preprint. Now that was membrane seg, and now we'll quickly show you what we're planning to have soon for membrane pick and membrane stats. So with membrane pick, the idea is that we want to be able to localize the membrane proteins. And you may have wondered why it's membrane V2, because there was also membrane V1, which was a pure membrane particle picker, but it did come with some limitations. So we didn't design it in a very easy to use way. I think that was a huge bottleneck, but it had some nice features. And one of them was that it was very annotation efficient. So even with a couple of training membranes, it already gave a decent performance. But the performance was also limited due to some design choices. So we tried to overcome that. And that's what we tried to do with MembranePick. So we wanted to improve the performance that the model can really have in localizing membrane proteins. But at the same time, we also wanted to keep this nice feature of the annotation efficiency. So just need a couple of training membranes to get a decent performance. And the main important thing, I think, is that we wanted to make it just much easier to use. So what we wanted to do is really have it head-to-head with an annotation software, so you can really easily go back and forth between the training and the annotation of membrane proteins. And that's what we did. So we got this better in-plane context to improve the performance by learning directly on membrane meshes instead of using the volumetric representation of the data with the DiffusionNet architecture, and we integrated it with an annotation software, which is Surfarama, which I will also show in one of the next slides. But that worked really nicely. So here's just a short glimpse in how it worked. We tried to evaluate how good the performance is when we trained both membrane V1 and MembranePick with a different number of training membranes. And it's nice to see that MembranePick already outperforms membrane V1 consistently, and it has this nice property of data efficiency again, where even with a single training membrane we get a reasonable performance out of it. And here on the right you can see MembranePick being applied to one of the data sets that we used. And the nice thing about it is, I think, we're still working on improving that, but I think it's already much easier to use than the previous version of Membrane V1. And we integrated that with an annotation software, which is Surfarama. And that was a nice project that we did in a hackathon last year together with Ricardo, Kyle, Lorenzo, and Kevin. And if you're maybe familiar with Membranorama, then this is pretty much doing the same thing, but it's just much more accessible because we implemented it as a Python Napari plugin. So you don't need a huge Windows machine anymore with strong GPUs, but you can ideally just use this Napari plugin and then click a couple of points. So what happens here is that you take a mesh representation of the membrane, and then the densities of the tomogram are projected onto the surface of the membrane. So you can really see where the proteins lie within the membranes, and then you can just click a bit and generate some nice ground truth that you can then use to train a MembranePick model. Right, and once you've successfully done that, you can maybe apply the MembranePick model on your data and then feed that into MembraneStats. So that's what we tried here. In this case, we trained MembranePick just on this left data set, which is thylakoids in spinach tomograms. We tried to localize photosystem II proteins within these. And that worked really nicely. So we got a good performance on this data set. But what's really cool about this is that we could use the same model that we trained on the spinach data set and apply that on two different data sets, also depicting thylakoid membranes showing photosystem II proteins. But this time the tomography setups were different. And also, it's chlamydomonas instead of spinach. And it's nice to see that the model is picking up some reasonable stuff. It's not perfect yet, but still it's a good starting point, I think, to get some further annotations on these other data sets and then fine-tune the model towards these. And you can apply MembranePick on these data sets and get some nice picks out of it, which then you can analyze in MembraneStats. So in this case, we just extracted some nearest neighbor distributions where we can see, between the different data sets, there are some small differences. And the same also for the protein concentrations here on the right. But MembranePick and MembraneStats, that is hopefully out to easily use very soon. But yeah, since membrane seg is the main part that is currently out there, I will focus on that from now on for the rest of this talk. And we saw already what's working in the background, but now I would like to go into just the practical use of membrane seg and some commands that you might be aware of, but maybe also not. So I will just dive into how that works. So you can always check out our documentation on our GitHub repository here, where since yesterday there is also a tutorial notebook that you can just open in Google Colab and then just click through the different cells, which basically shows also the different steps that I'm showing here. So all the steps that I'm showing here, you can just easily reproduce them in this notebook by just clicking a couple of cells. And yeah, the membrane seg program is basically just a command line, a collection of command line commands, with this one being probably the most important one. Because we try to have this out of the box segmentation. Ideally, you would just type into your command line membrane segment and then pass the path to your tomogram and the path to your checkpoint. So the path to the checkpoint, we always provide the latest checkpoint that we have on our GitHub repository, but also older ones, so you can try to reproduce the old performances. But yeah, always keep an eye on the GitHub repository to get the latest checkpoint. And then you can really just feed in the tomogram in an MRC format, and you will get out the segmentation in an MRC format as well. So yeah, ideally that works already nicely out of the box. But if not, then there are also some other commands. So you can see what are the different commands that you can choose from by just typing membrane segment. And that will show you this list of different commands that you can really use to optimize the performance of membrane seg. So the most important commands are, of course, these two that I just showed you, the tomogram path and the checkpoint path. So these are also obligatory. And all of the others are just optional commands. There's some easy stuff, of course, just defining the out folder where you want to really store your predictions. But maybe a bit more interesting is this rescale patches functionality, which is relatively new in membrane seg, so maybe you're not aware of that yet. And it's basically doing what it says. So, I mean, we trained membrane seg on a specific pixel size, which is roughly in the range of 10 angstrom. So if your tomogram deviates a lot from this pixel size, then the performance of membrane seg will probably be a bit limited. So it may make sense to rescale your tomogram to this specific pixel size that we trained with, or at least get in a similar range, and then apply the model. And this rescale patches functionality does that basically on the fly during the prediction, which I think is a relatively elegant way of doing that. So what happens normally is that if you use this command without any flags, then we have this sliding window approach where we extract these small patches of the size 160. We feed them into the model. We get some segmentations, and then we put them into the respective position in the output segmentation. Now, if you activate this rescale patches flag, then we will extract a patch of a different size. And the size of this patch is determined by what happens if we rescale it. So we'll extract this patch in a size that if we rescale it to 10 angstrom, it will also have this size 160. So we extract this patch, we rescale it to 10 angstrom, get a smaller patch of size 160. We can feed that into the model and get some good segmentations out of it, and then rescale back to the original size, and then put it into the position of the tomogram, the output segmentation. Now, of course, you can also alternatively just take your full tomogram, rescale it to 10 angstrom pixel size, and then feed it through the model and get a segmentation in 10 angstrom, and then rescale that back. But I think this approach here can be a bit more elegant because this is implemented pretty efficiently because everything in the rescaling is happening on the GPU. So this does not really cost any time of the rescaling. So yeah, if you have tomograms in a different pixel size, then I would highly recommend to try this rescale patches here. And important arguments for that is, of course, the input pixel size, which normally membrane seg reads from your header. But yeah, headers can be corrupted. So I don't know, some programs set them to 0 or 1. And if that happens, then you will get some problems with the rescaling. And out pixel size, that's basically where you want to rescale the patches during inference. So normally by default, it's to 10 angstrom. So I would not deviate too far from that, but that's also something that you can play around with if you're not happy with the performance. Now, two other functionalities that might be interesting is this store probabilities and store connected components functionalities, where the store probabilities output the score maps of the network before thresholding. And this, I think, can be really useful to get a better understanding of what the network actually does. Because otherwise you just have this binary segmentation and it's performing okay, but you don't know why it's performing that way or why it has the problems. And these score maps, they can give a bit more insights on what's really happening. The store connected components on the other side, that's more of a post-processing thing. So once you have the binary segmentation, this will try to assign different integer values to different instances. So if you have completely separate membrane instances, then this store connected components functionality will assign a different integer to both of these instances. Whereas normally the output would be just binary, so just 0 or 1. So here this is visualized again. So this is just the binary output segmentation, which is the default output of membrane seg. But then if you activate the store probabilities, then you can get these scores before thresholding, and you can see, get a better feeling of what the network actually learned. And the nice thing about this is now you don't have to necessarily stick with our default threshold, which is at 0. So you can just choose an arbitrary threshold that looks the best for you. So maybe you want to reduce some noise, then you can choose a bit of a higher threshold, or you want to increase the membranes that are segmented, then you can go for a bit of a lower threshold. And if you choose the store connected components flag, then maybe this is not the best example because here in these tilecoids everything is kind of connected. But yeah, ideally, you will get a different integer assigned to these different membranes. So, for example, if you have a huge, if you have a lot of vesicles floating around or something like that, then each of these vesicles ideally will get a different integer assigned to them. And the last thing that I wanted to show you is this test time augmentation. So maybe if you've used membrane seg already, then you saw this bar here running through 8 times, and that's exactly this test time augmentation. So what happens is that during inference, we don't only feed in the tomogram into the network and we get out the segmentation, but we also feed in the tomogram, we feed the flipped version in X, flipped version in Y, and in Z, and also all different combinations of flips and Y and Z. And we feed all of these different combinations, which is in total 8 combinations. We feed them into the model, and then we combine the output of these with the intention that that this just makes the predictions much more robust. So I think this will improve the segmentation performance by a bit. And I would normally recommend to use that, but if you're really after a very quick segmentation, or you have to segment a big data set, then maybe you want to turn this off by setting the no test time augmentation flag, which will speed up things by a lot because you just run through this bar once instead of 8 times. So these are the main functionalities of the membrane segment command. But there's also some more functionalities that I quickly want to show. So you can get an overview of what membrane functionalities are available by just typing membrane. So here you will see this list again of different functions that you can perform that. And membrane segment is the function that we just discussed in detail. If you just type that, then you will get to the interface that I just showed you. But then there's also other stuff. So the components and threshold, that's very close to also what I showed you before. So the components, in this case, is just post-processing where you would feed in the binary segmentation and then get the connected components labeled with different integers. And the thresholds functionality that works very closely with the score map that you can output. Because once you have the score map, you can use this thresholds functionality to just set up a number of thresholds that you want to try maybe for your segmentation. And then this will just threshold the score map at these different values and output the segmentation so you can easily compare and choose the best threshold for you. Other functionalities, there's the training, of course, so that you would be able to train your own model. The thing about that is that, unfortunately, we haven't released our full training data publicly yet, which will hopefully happen soon. But once that is out, I think it should be very easy to just take the full training data set, combine it with some of your data, and then really train a nice generalizable model that is performing even better on your specific data. In the meantime, there's these fine-tune functionalities, which not necessarily require to have the full training data available. In theory, it should be able to perform already reasonably well with just a couple of annotated patches from your specific data set. And then we applied some heuristics, and you can train this model with this functionality and ideally maximize the performance on your data set. So we provide a couple of documentation on our GitHub repository how to properly do that. This is still a bit experimental, so if you want to give it a try, I'm always happy to get some feedback on how it works for you. And now, as another functionality, what might be interesting is the skeletonization function. Because sometimes if you want to do some downstream analysis with these segmentations, then often the format that is required is not the full volumetric segmentations, but often the packages require some skeletonized segmentations. So really just these one voxel thick sheets. And that is what skeletonization, this membrane skeletonized function basically does. So it takes as input the binary segmentation, which has some thickness to it. And then we implemented the algorithm from TomoSegmentDB to really get a 3D surface that is just a very thin sheet. So ideally, it's really just a one voxel thick sheet that you will get out of this. So that's about the functionalities. Some pre-processing, that's the last thing I want to show, because sometimes your performance with membrane-seg may be not ideal, and that may be because the tomogram is looking a bit differently to what our training data looked like. So in this case, we provide this Tomo pre-processing functionalities that you can access, again, just by typing Tomo pre-processing, and then we have some functionalities. So for example, the match pixel size functionalities, which is basically what I explained before, the alternative to the rescale patches functionality. So in this case, you would just take as an input the tomogram, rescale that to another pixel size. Then you can feed that into membrane-seg or do whatever you want with it. And then rescale the segmentations back to the original pixel size again. So I think this can be useful in some cases. And another thing that may be useful is also this deconvolution filter that is also very similar to the one that is implemented in warp-M. Because sometimes, if the contrast in your tomogram is a bit too low, then membrane-seg can struggle in some situations, and it may be beneficial to just increase the contrast of your tomograms to get a decent performance out of it. Right. And I think that's the main functionalities that I wanted to show. Short advertisement again for this tutorial that we created a couple of days ago, so that you can find on our GitHub repository on the Team Tomo GitHub. And this will guide you through basically the steps that I showed you today. And hopefully it's easy to use. Here again, you can find it here via this barcode. You can get to our GitHub repository, and I think with that I'm already done. So I just want to thank everybody involved in this project. So that's the Peng Lab here in Munich, but also of course the Cell Architecture Lab with Ben Engel in Basel, and everybody, all the collaborators that were involved in this project. And of course, thanks a lot to you for your attention and for listening. So yeah, let me know if you have any questions.
Great, thanks. That was a really good talk. It's a really powerful piece of software, and it is very user-friendly in my hands, at least, given that I have no idea what I'm doing with tomography software most of the time.
That's great to hear.
So
Thanks.
while we wait for a couple of questions to come into the Q&A or the chat, I guess I'll kind of lead off with one based on what you were saying at the end. So you were saying the performance is sometimes improved by running deconvolution in that kind of warp-M style. Are there any issues if you go one step further and you do, say, a full denoising on your tomogram? So you get one of these kind of noise-to-noise style denoised outputs. Does it make it better or worse?
Yes. So in theory, I would always recommend if you have it available, I would always go for denoised tomograms. So ideally, CryoCare denoised tomograms. I think that is the best performance that we observed so far. I think it's just making the job a bit more easy for the network. And also, some of our training data is already denoised. We try to get a mix of raw tomograms and denoised tomograms, but I think in general, it's easier for the network to learn from the denoised tomograms, and therefore also the prediction performance is better with these.
Okay. Kind of related again is these missing wedge augmentation packages like ISONet. Do those also help, or do those introduce artifacts that mess with the segmentation?
Yeah, we haven't explored that into very much detail yet. I would say as soon as it's visually getting some membranes back, then it can definitely help. We also try to imitate this process of ISONet kind of with these missing wedge augmentations that we performed, and we tried to replicate basically this missing wedge filling in that ISONet does. It's working sometimes it's not working so well. So yeah, I would say it's not necessarily helping with ISONet, but it can definitely be worth a try. I think especially for vesicles, I saw some really good performance if you have kind of spherical shaped membranes. Then I think ISONet also often does a good job of restoring these densities, and then I think this is also very beneficial for membrane seg.
Yeah, that's what I had noticed doing ISONet in samples which had like virus-like particles in them, is that after running ISONet, membrane seg would then pick up like these VLPs as if they were vesicles, because it restored the spherical nature. Okay, so we got a question from Abigail Watson. They've been using membrane seg and really enjoyed it. For the training patches, they've noticed there's a frame with ignore pixels around the edge. Why is there like a solid ignore frame around boxing around the tomogram, and is that a standard size, or does it vary based on the sample?
So yeah, we always put this ignore frame around the patches for multiple reasons actually. So the first reason is really a practical one. So first of all, it's always the same thickness. So we always use I think a thickness of 16 voxels towards the edge and use that as the ignore label. And one thing is that it's just often very hard during manual annotation to really know what's happening, because we just look at this small patch and we have the context from one side, but we of course don't have the context from outside of this patch. So we would have to go back to the tomogram and look what's really happening there to get some reasonable manual annotations in there. So for the manual annotations, this can be a bit tedious to get some proper annotations in there. But the same problem also exists for models. So if the model doesn't really see the context from outside of this patch, then and you tell it there is a membrane and it doesn't really have this context to know where this decision came from, then it may also lead to some distortions. So we just went very easily for removing that, so you can see as a manual annotator, okay, this is the context that I need, and also the network sees exactly the content that it will need.
Yeah, that makes sense. Okay. While I wait for a couple more questions to come through, I had a question on the particle picking segment, which I guess isn't released yet, but I guess the two questions I have on that is, are you I noticed you're using Photosystem 2. Are you benchmarking on, say, a variety of different sizes of membrane proteins, or are we talking, does this only work on large systems like FATPAs and things? And I guess the other part is, how are you handling downstream use of the coordinates? Like, are you generating star files or warp boxes to like pass off onto the downstream subtomogram parts?
Yes. Yeah, for the benchmarking, we definitely try to explore on which proteins it works. Problem is again the ground truth availability. So for the Photosystem 2, we had some projects on our own. We have some nice ground truth data sets, so we can easily compute some statistics, and we can see, okay, it's performing well. But we also, we ran it on a couple of other data sets as well, showing different proteins. I mean, easiest, of course, ribosomes or some phycobilisomes that we will try to include in our evaluations, and there it worked pretty nicely. So I would say that as soon as you can see the membrane proteins properly, then membrane-Pig can do a nice job of picking them up. I mean, it will never do some magic and just magically come up with some particle positions that you cannot even see visually. But I think normally, as soon as you can see it properly, then membrane-Pig, from our experience, does a nice job in that. And regarding the output format, I mean, yeah, membrane-Pig, currently it's only outputting membrane protein positions, so we don't have at least in-plane orientations yet. So what we do is that we take the positions, we put out Euler angles in the to basically align the membrane for all the proteins. That's what we can do, but then we don't specify the in-plane orientation. And the format of that currently is that we use some Reliant format, but yeah, some Reliant star file format, but also very happy to implement other output formats of that.
Okay, cool. That's because particle picking in tomography is still extremely painful. So particularly with membrane-bound proteins. So if you can get something where you can specifically pick your membrane protein and not the other 400 membrane proteins that are also in your sample, that would be very helpful.
Yeah, this is still a bit tricky. I mean, the one thing about membrane-Pig is I think we're definitely not there yet that we have this one general model like for membrane-Seq where we can just say, okay, feed it through membrane-Pig and you will get some nice positions. So you will probably, it will be very specific to the problem that or the project that you're encountering right now. So if you look at the membranes, then you would need to annotate manually a couple of proteins at least that you're really looking for. And then ideally it can generalize nicely to the other membranes that you have. But yeah, it's a general model. I think that would be super cool, but unfortunately we're not there yet.
Yeah. Yeah, I think that will be that's a much more difficult problem. I don't know if we'll get there with tomography in the same way that we did with single particle for the particle pickers. Okay, we have another question in the chat, so I'll just take one or two more from the chat and then we'll wrap this up because we're coming up for the hour. So, would a very distinct bilayer impact the skeletonization process?
A very distinct bilayer. So basically, if you can properly see the bilayer in the tomogram, if I understand it correctly. So I mean, ideally, if you have the nice bilayer of your membranes, then membrane-Seq would be able to, or would by default not segment each bilayer separately, but just give you one segmentation. And then once you have that, the skeletonization is just a post-processing that will take this binary segmentation and not really care about the bilayer. But yeah, of course, if you have two membranes that are very close to each other, then or two segmentations that are close to each other, maybe because membrane-Seq did a bad job of segmenting them as one, but instead segmented each bilayer separately, then you will probably also get separate skeletons for each of those.
Okay. Oh, so the expected result of that is for a tight bilayer, you would expect and hope to only get a single segmentation.
Exactly, yes.
Okay. Great. Okay, well, I think we're wrapping up most of the questions that have come through so far. So I'd just like to thank Lawrence again for a really good talk and a really extremely helpful tool for dealing with tomography data. And I'll just check that next month we are coming back with a talk from Schrödinger about Phoenix and Glide-EM for enabling cryo-EM structures for drug discovery in Schrödinger. So if that sounds interesting to you, please come back next month. And with that, I guess we'll just wrap this one up. So thanks, Lorenz. That was really great.